Welcome back. We were discussing in the last session the different types of viruses, and we have seen that viruses can be classified into DNA, primarily into DNA virus, RNA virus and retrovirus. DNA virus, the DNA is the genetic material that is injected, that goes into the nucleus ultimately, and then either it could be integrated or it could be remain as separate entities, but then it utilizes the viral this host machinery, that means all the DNA polymerase and the your RNA polymerase that is utilized to make the mRNA corresponding to the virus, and then the mRNA synthesizes the proteins, the virus particle is generated and comes off from the cell. In the case of RNA virus, there are two types. We ended up by saying that RNA virus, the life cycle depends on whether it is a negative sense RNA virus or a positive sense RNA virus. What is a negative sense RNA virus? That means the mRNA has the sequence which is complementary to the actual mRNA that is required. You have to understand this. Negative sense, negative strand RNA virus, that means the RNA has the sequence which is basically the sequence of the negative strand of the corresponding DNA. If you think of the DNA, although it does not have the DNA, the negative strand mRNA means it is not the correct sequence. Now, you have to, when you copy this, you have the complementary bases that are taken up. So, from negative strand mRNA, if there is a RNA polymerase, then whatever you get, like this is the your genetic material, and now the genetic material is suppose this, but if it is negative strand, then when you copy this, what you will get is the actual mRNA. Positive strand means the actual one, the sequence matches with the RNA that is required to synthesize the proteins. Okay. So, now that means in RNA virus, if it is a negative strand RNA virus, so basically you do not have to go to the DNA, you directly copy it to the mRNA which containing the correct sequence, and that mRNA makes whatever proteins are required, and then that is packaged and finally that comes off. That is the negative strand RNA virus. But if you have a positive strand RNA virus, then the job is little bit more. No? You have a positive strand RNA virus. So, first make a negative strand of that, and then negative strand is copied back. Okay. So, you get the actual mRNA. Okay. But here this does not go, it is not required that this virus needs to go to the nucleus, because it is not, the DNA is not involved anywhere, okay, in the RNA virus. So, it takes place in the cytosol, that means outside the nucleus, but within that within the contents of the cell or the contour of the cell. Okay. So, that is what is RNA virus. And now, so there are two types, plus and minus strand. One scientist, a biologist, famous biologist, David Baltimore, and he first proposed this classification. Baltimore's classification, I am not showing it, but you can get it from anywhere in the textbook or in the internet. He actually classified into 8 types, but these 8 types are basically, I told you, the DNA could be double strand, could be single strand. So, there are one classification there. Then the RNA could be single strand, double strand. Then the RNA could be negative sense RNA strand, positive sense RNA strand. Okay. And then you have retrovirus. We have not discussed the retrovirus. So, you see, you can get 8 classifications. That is the famous classification by David Baltimore, a Nobel winning scientist, biologist. Okay. Now, let us come back to the retrovirus. I think I can, if it is not there, I can tell you what is actually happening in retrovirus. In retrovirus, now again you have this RNA as the genetic material, but so RNA as the genetic material. Now, as it enters the cell, again like the DNA virus, this RNA is injected inside the nucleus, okay. And then this RNA gets this RNA is the RNA is copied into the DNA first. That is the reversal of the transcription process. What is transcription? DNA goes to the RNA, because DNA is transcribed to the RNA, but this is a process where RNA is copied back into the DNA. Okay. So, this is the DNA, the viral DNA. Okay. Now, there must be some enzyme. We do not have this machinery. Our system runs from DNA to RNA to protein. Okay. So, we have transcriptase, means the polymerase, the normal polymerase, but this is a reverse of transcription. So, this enzyme is called reverse transcriptase, reverse transcriptase. Okay. So, this DNA is now entering into the nucleus, and then it will be integrated into the host DNA. So, there is a question of integration here. So, if this is the host DNA, so now you have the viral, so you have the viral DNA. Okay. So, what happens again, just repeat. What is the difference between retrovirus and RNA virus? RNA virus enters and it has its own machinery. Okay. It does not need to go to the nucleus, directly it is copied the RNA to the mRNA. Okay. And but for retrovirus, the RNA has to be copied into the DNA, the DNA then goes into the nucleus and then integrates with the host DNA, and this then is utilized for the information flow. So, when the information flows from this part, you will get mRNA of the virus. Okay. mRNA of the virus, that is what you need. What the virus needs to replicate is to have the mRNA, and once the mRNA is formed, then new protein particles will be made, and with the proteins which are required for the virus replication. So, the virus particle will be formed and then it will come out of the cell and finally infect another cell. Okay. So, that is how the infection goes. So, now we know how to what are the different types. Now, the question is how to treat a patient who is suffering from viral infection. One thing is that vaccination. Usually, originally what happened for viral infection, people realized that many of the viral infections are cured by taking rest for 1 week. If you take any medicine, you will be cured within 7 days, and if you do not take medicines, it will take 1 week to get cured. That means, it is same without medicine, with medicine, it does not matter. So, basically what ultimately makes me cured if I am suffering from a viral infection? Usually it is a cold, some flu type symptoms I have, and basically what happens, that immune response, the immunity in the body, that ultimately takes care and takes care of the this virus particles whatever is in my body, and then ultimately destroys them. So, basically it is the immunity that is very important. Now, the problem is, if a person has less immunity, then what will happen? Then you have to provide him, then vaccination. See, vaccination is something which is basically boost up the immune function. Means, if you have a virus and if you take a dead virus of that, inject it. So, what will happen? Some proteins will be, some antibodies will be generated, antibodies are proteins, and that will be there, the memory which has a memory. So, whenever the next virus particle enters, so it immediately knows that this is the type of virus particle, and then immediately you can the body will take the will make these antibodies and destroy the virus particles. So, vaccination is the general route for protecting against the viral infection, and there are different vaccines which are very successful, polio vaccine, measles, mumps, MMR. As this MMR vaccine is a very standard which is given to the babies, and then however, not all viral infections you cannot that there are vaccines. The vaccines are basically discovered by way back about I think 300 or about 300 years ago by Edward Jenner. He developed the vaccine for the pox, for the smallpox vaccine. At that time nothing is known, nothing was known, science, only physics and mathematics came into force. Chemistry started by discovery by Lavoisier and other people of the different gases, oxygen, hydrogen, nitrogen. So, nothing was known, but Jenner, just by sheer observation, what he observed, that the milkmaid, means who actually trades with the milk, okay, they are generally suffer, the milkmaid who had contracted the less virulent cowpox. They actually have some type of pox, but that is not fatal. Okay. So, they have some blisters in their hands, and that what Jenner noticed, and later on they never had any smallpox. So, those persons who are milkmaid, so that means this is what is called cowpox. They are handling the cow and they get a pox which gives blister, but that is not life threatening, okay. But that protects the person from getting smallpox, which is life threatening for the humans. Okay. So, what he did, he took the whatever contents inside that blister and then injected into the other people, and that is the start of the vaccination. Okay. However, as I told you, vaccination works as you told, that you have to either dead virus you can take or a part of the virus particle you can take, so that antibodies can be generated. But the problem is some of these viruses like HIV, which causes the AIDS epidemic, and then the flu virus, some of the flu viruses are very dangerous, bird flu or then Nipah virus. There are different types of viruses are there, swine flu. So, for them it is very difficult to have a vaccination because of two reasons. First reason is very important, that is the virus changes its character, there is mutation. So, if you want to see that today the virus looks like this, and then you generate a vaccine, and the next day you see the virus has changed, the surface has a different characteristics. Okay. So, that vaccination will not work. So, that is the mutation problem. So, you have to have, the reason I am saying all this, that to stress the fact that we need antiviral drugs. Okay. Now, the question is, how do we now get the antiviral drugs? I told you it took a long time to come up, because people thought that antiviral drugs will be very difficult, because it is utilizing the host machinery. Okay. But still there are certain differences. What are the differences? If you take suppose a DNA virus. Okay. Now, DNA virus, what happens? See, I have normal cells and I have say virus infected cells. Suppose this is virus infected. The virus infected cells, that means the virus will generate what? Will inject its DNA into the nucleus. If it is a DNA virus, what is the DNA virus? Like the blisters that we get in the lips, which is caused by a virus called simplex herpes virus. Okay. So, that is the DNA virus. Now, what happens? Sometimes they may be a little painful and also cause lot of uncomfortable, bring in you do not feel comfortable having this simplex herpes virus. They also can infect the eye, that is herpes infection in the eye. So, you need to treat this herpes infection is DNA virus infection. Now, what happens here? The DNA is injected into the nucleus, and then the DNA the DNA, there are two ways I told you. It could be integrated into the, it could be the integrated into the DNA, or it could just remain there and then it will utilize the host machinery, the polymerase, and then make the mRNA. Okay. Now, how the polymerase works? The polymerase works by, again coming back to that, just going back to the biochemistry, that the polymerase works by attack of the 3' OH into the, to a 5' triphosphate, 5' triphosphate, and then form the phosphodiester bond. So, this phosphate is attacked, you kick out a diphosphate. So, that is the reaction we are talking about. Okay. So, now the DNA, that is, suppose we talk about the DNA which is not integrated, which is like this, okay, which is released but not integrated with the DNA. Now, what will happen? This DNA has to be copied. Okay. And if you want to do that, suppose I take a compound which is, there is a drug called acyclovir, acyclovir, actually that is the pronunciation. Now, what happens? The acyclovir, you know that the one of the component of the increase of the polymerization reaction is the attack of the, is the triphosphate, is the nucleoside triphosphate, that is one of the component. Okay. So, what you have done, suppose you take a nucleoside which suppose does not have the 3' OH. Okay, the base is there and you are giving it in the form of OH, and you have a group here X which is not able to attack the phosphate here to make the phosphodiester bond. Okay. If that be the case, then what will happen? If this is, of course, this is not given as the triphosphate, it is given as the only the alcohol, that means only the nucleoside, but to participate in this polymerization reaction, this has to be transformed into the triphosphate. Okay. So, what I have said, that suppose I want to stop this process, that means DNA going to the mRNA of the virus. So, what I need to do, I need to add during this transcription process, I need to add a nucleoside, but not the triphosphate, again I repeat, a nucleoside which lacks the 3' OH. That means, the 3' OH cannot any further, cannot continue that reaction, the phosphodiester bond formation. Okay. Now, the first drug that was made on this principle is this acyclovir. So, acyclovir, again, even if I write now, you see O and then you have a base. I am just not writing the adenine, this is guanine. Okay. So, I just wrote the B. Now, what will happen? This is basically a truncated version, of the sugar, but you do not have this part, but it is a truncated version of the sugar. Now, what happens? Interestingly, this is also taken up for as a kind of nucleoside triphosphate for lengthening the chain. Okay. But what happens if this is taken up by the polymerase, then the next reaction, the polymerase basically have, if this taken up as a base, then what will happen? The sugar that is here, that lacks the 3' OH, lacks the 3' OH. Okay. So, if it lacks the 3' OH, the chain cannot grow any further. Okay. So, chain will be stopped. The question is, why this will be taken as a or how this is taken as a substrate for the polymerization, when it is not present in the triphosphate form. Okay. You have given it only in the alcohol form, free alcohol. So, in order to have this taken up by the polymerase enzyme, recognize it, it has to be transformed into a triphosphate. Okay. Now, here is the interesting part. You have two cells, one cell is virus infected, another is the normal cell, no virus. Okay. Now, this acyclovir molecule enters both the cells. Okay. Now, the virus has now the it has to be converted into a triphosphate. The triphosphate is written here, but the triphosphate takes place in in stages. Okay. What are the stages? First, it will undergo monophosphorylation, and then it will undergo diphosphorylation, finally triphosphate. Then that will be taken up as the substrate. Okay. Interestingly, the first phosphorylation is done by a viral kinase. A viral kinase. Okay. We do not have any, our body does not have any kinase. Kinases are basically they make the phosphate, they does the phosphorylation, they do the phosphorylation. So, this but viral kinase, we do not have any corresponding kinase to do the monophosphorylation of this. Okay. So, once this is monophosphorylated, then the host kinase takes up the further phosphorylation. So, it makes the diphosphate, then the triphosphate. So, what happens when this molecule enters the two cells, one is infected, another is uninfected. So, the infected cell, there is viral kinase present. So, that will be converted into the monophosphate, and if it is converted to monophosphate, then only the host cell machinery converts it to the triphosphate, and if it is converted to triphosphate, it is taken up as a substrate by the RNA polymerase. If it is taken up as a substrate, then what happens? As it is incorporated into the chain stops from further elongation. So, the mRNA will be truncated, will be a truncated mRNA. So, it will not be a proper mRNA. Now, there are two mechanisms by which this acyclovir can work. One is that it makes the triphosphate via this process, the viral kinase followed by the host kinase, but it either it acts as a competitive, it acts as an inhibitor, it goes and binds to the RNA polymerase, stays there, or it can react to form the phosphodiester, but after that the RNA cannot grow any further. So, there are two ways it can stop the RNA polymerase. One is acting as an inhibitor, binds to the active site, and the other is it is actually incorporated into the growing chain, but once it is incorporated, the chain growth stops. Okay. So, basically virus cannot make the mRNA. So, that is the first landmark discovery that an antivirus drug was discovered. Okay. So, that means you can, if you have very good biological knowledge about the virus, the life cycle, you can make compounds which are antiviral compounds. Basically, you have to identify the processes which are different from the host. Now, problem with acyclovir is that it is not very, it has got some bioavailability issue. Okay. It is not absorbed properly from the gastrointestinal tract. So, see, it has to be absorbed and then it goes into the bloodstream and then attack the cells which are virus infected. So, to have that absorption better, what people have done, they have put an ester which is an ester of valine. Okay. Now, valine has a recognition. Valine is a natural protein amino acid. So, there are receptors to hold the valine part. So, in the GI tract, the valine is recognized. So, that is recognized, taken up, and the absorption is better. Okay. And then that goes into the bloodstream. So, these are nothing but prodrugs. See, all are prodrugs. If I say what is acyclovir, that is a prodrug, because it has to be converted into the triphosphate before it actually stops the RNA polymerase. Okay. So, it is a prodrug, all are prodrug. This is basically another step ahead of the before the prodrug, because you have to cleave the valine ester bond and then you have to make it triphosphate. Okay. So, that is the DNA virus. Similarly, now people have developed compounds which are anti-HIV drugs. What is HIV? HIV is a retrovirus. HIV is a retrovirus. Okay. So, in retrovirus, again I remind you, in retrovirus what happens, that you have to integrate the DNA which is obtained by reverse transcriptase process from RNA to DNA, and this DNA gets integrated into the host. So, here that is what is retrovirus. Okay. Then as it is integrated to the host DNA, so the host DNA will copy to the mRNA. Here one new enzyme that is there, that is called reverse transcriptase. Okay. What is reverse transcriptase? Reverse transcriptase is making of the DNA from the RNA. RNA is the genetic material, goes to the DNA, and that is what is done by the RT, reverse transcriptase. But this is nothing but making of DNA means what? It is a polymerase. It is a polymerase, but it is an RNA dependent DNA polymerase, because the template is RNA and you are making a DNA. Yeah, you have to be careful about naming all these. If I say RNA dependent DNA polymerase, that means I am making a DNA utilizing an RNA template. So, that is a reverse transcriptase. And what is a transcriptase? It is a DNA as a template, but you are making an RNA. So, that means it is a RNA polymerase. Okay. I think it is clear. Now, this reverse transcriptase is because it is a polymerase again. Polymerase means you have to make this phosphate, that phosphodiester bond. So, what people found, that the same strategy worked for HIV. The first drug that was made was called zidovudine, or it is popularly known as AZT, azidothiamidine. Okay. What it has? It has got a thymidine here, thymine base, and it has got, it lacks the 3' OH, but it has got an azide and it has got the OH. But the basic principle is same, that if it is taken up as a substrate, chain elongation will not take place. Now, the question is, how does it, how does the triphosphorylation works? Like in the earlier acyclovir, it is the viral infected cells, the monophosphorylation done, and then the diphosphate and the triphosphate that is formed by the host kinase. In case of AZT, contrary to the earlier case, here if you have two cells, suppose HIV infected and HIV non-infected, okay, this AZT enters. Here the entire phosphorylation is done by the host kinase. Okay. So, apparently it seems that why it will be very specific, because if the triphosphorylation is done by the host kinase, so if this AZT will be converted into the triphosphate in the normal cell as well as in the HIV infected cell. But fortunately, this reverse transcriptase, this triphosphate, what is made out of AZT, has got much higher affinity for the reverse transcriptase than towards the other polymerase enzymes that are present in the host. I hope this is clear. Here first of all, there is the difference in mechanism. Here the phosphorylation is done by the host kinase. Okay. So, phosphorylation will be done in both the cells, infected, non-infected, but the triphosphate that is made up from the the AZT is recognized by reverse transcriptase much more than the transcriptase or other polymerase enzymes that are present in the host. So, basically what will happen after the triphosphate formation, it is the HIV infected cells that will be affected. The AZT will work against the HIV infected cells, and where it is having the phosphorylation, that will ultimately come out of the cell, because it does not have any affinity. Means it has got affinity, but there is a huge difference between the activity of affinity for the reverse transcriptase vis-a-vis the polymerases that are present in the host. Okay. So, that is the mechanism of AZT. On the same line, you have got different drugs that are, first drug was AZT, then somebody put a sulfur here, then somebody removed the sulfur, nothing is there, only dideoxy, it is a dideoxy system. Okay. And a different base also, you can have different bases also. So, there are many permutation combinations you can get, and there in fact, there are lot of reverse transcriptase inhibitors now available, okay, for treating the HIV. You know, AIDS is a deadly disease. What it does? It destroys the immune system, and ultimately the patient dies of very opportunistic infections. Some infections that we have, those are called opportunistic. Opportunistic means they are all waiting to invade the body, but because of the immunity they cannot do that. So, when the immunity goes down, these opportunistic microorganisms go inside, and then ultimately, because immunity is not there, ultimately nothing works. So, the person dies of that. Okay. So, basically these are called like the hyenas, they are opportunistic animal. No? Hyenas never kill any other animal, it is the lioness kills the animal, and then the hyenas come and then try to take over whatever remains are there, remove that, kick out this lioness, because they are large numbers, and then the lioness leaves and leaving the dead animal, and then they eat that dead animal. So, they are opportunistic animal. So, you have similar opportunistic infections also. So, this is one way, reverse transcriptase inhibitors. Now, reverse transcriptase inhibition can be, reverse transcriptase is an enzyme, it must be having some active site where this dideoxy or the azidothymidine triphosphate is binding, okay, and then the polymerase works. So, basically the AZT triphosphate has to bind in the active site, then that will be incorporated into the growing chain. Now, there is a process which is called allosteric inhibition. If you remember, I told you allosteric inhibition is basically that the inhibitor binds to a different site, but when it binds to this site is closed, the normal site, that is what is called allosteric inhibition. So, basically it is a non-competitive, because they are not going into the same active site, the normal active site is there and there is an allosteric site. So, there is a molecule which goes here, it does not bind here, but as it binds here, this binding pocket is closed. So, that is also possible, you can have allosteric inhibition of reverse transcriptase, and one example is shown here. Let me see whether the structure, no, that is not the one, sorry, I think I will yes, nevirapine is a drug which is an allosteric inhibitor of reverse transcriptase. Look at the structure, it is also called a non-nucleoside inhibitor, because earlier AZT and all these things are nucleoside, base and sugar, but now you have a non-nucleoside inhibitor, that is the nevirapine is one, maybe some more new ones have come up, okay. Because the treatment of AIDS, because the virus changes the characteristics, it mutates very rapidly. So, you have to target the virus by different techniques. If you give the same type of molecule like only AZT or other nucleoside, it may not work in the long run. So, better you have a cocktail of drugs, that one targets the active site of this reverse transcriptase, another targets the allosteric site, and if there is another mechanism by which this reverse transcriptase can be inhibited, then better do that also, add that into the cocktail. Now, yes, there are again scope of making new antiviral agents for HIV, because remember HIV is a retrovirus. So, retrovirus, what it does? It has got a reverse transcriptase which is not present in the host, and I told you that the retrovirus, that RNA has to be copied into the DNA and the DNA has to be integrated into the host DNA. Okay. Who does the integration? That is also another process, because your DNA is, your host DNA is here. So, somewhere you have to insert your viral DNA. Okay. So, there must be some protein which is doing that, chopping at some point, putting the virus DNA here and then sealing it. Okay. So, that is what is called an enzyme called integrase. So, now you have a scope. You can inhibit the integrase also, that is possible. However, not much success is yet to be achieved by inhibiting the integrase, but it is a good target. But there is another target, and that is how the virus enters the host cell. How does the virus enter the host cell? I told you that there is a recognition point. There has to be a recognition on the cell surface and the virus particle. Okay. So, and this, whatever the envelope, that is made up of glycoprotein. Okay. Who makes that glycoprotein for the virus? Must be some enzyme that is present in the virus. Okay. And what was found was a protease enzyme, which is very crucial for the virus to replicate, to enter into the cell. Because this HIV protease makes the glycoprotein. See, initially the mRNA, the viral mRNA, what comes out of the nucleus is very big, because it has to make all the proteins. So, it makes the protein, first it makes a big protein, and then the protein needs to be chopped off into the actual functional components. Okay. This chopping, the big protein has to be chopped off. That means you have to need a protease to do that. So, the virus HIV protease is very crucial for the virus to amplify, to replicate, because if you don't allow this chopping to be done, then the virus particle will not have the glycoprotein, proper glycoprotein, which allows it to anchor onto the surface of a cell. Okay. Protease enzyme, which is very vital for this replication process of the virus, to make the proper glycoprotein. So, people started studying what is the structure of this HIV protease, and after the advent of this, now the X-ray crystallographic and the cryo-EM, all these are available to know the structure very quickly of a protein. So, HIV protease structure was solved, and it was found that it is a dimer. Okay. It is actually basically two similar units forming a dimer, and the active site is basically in the cavity that is there when the dimer is formed. So, basically you can dissect it into this. So, one part is subunit is here, another subunit is here, and you see there is an empty site here, which is the active site, and this is the flap region, and through which the molecule enters. Okay. Now, this is I think I might have a better picture of HIV protease. This is a crystal structure. You see, this is one part and this is the other part. So, it is basically a C2 symmetric dimeric enzyme which does cleavage of the peptide bond. Okay. Peptide bond, the question is, proteases we know are 4 different types. one is serine protease, cysteine protease, aspartyl protease, metalloprotease. So, first question is what type of protease is this? They found that this is an aspartyl protease. That means the aspartate, the aspartic acid and the aspartate, they are like this. Sorry, double bond O, this is not there, and OH. So, the water is held up by the aspartate. Okay. The aspartate removes the hydrogen, helps the water to remove the hydrogen from here, and then thereby increasing the nucleophilicity of water. Okay. So, that attacks the carbonyl of the peptide. So, it is an aspartyl protease. Again I just said aspartyl protease is basically one aspartic acid and one aspartate. So, first the aspartate takes up the hydrogen from the water. So, making OH minus, virtual OH minus, that goes to attack the carbonyl, hydrolyzes the kicks out the nitrogen, and then the OH minus, and then it has to put back the hydrogen to the carboxy, to the aspartic acid that is there. Okay. So, maybe I can it takes a lot of time, let us say. Anyway, I think that has been covered earlier, the aspartyl protease. Okay. So, two aspartic acid are there, one is here, another is there, and then the peptide bond that is hydrolyzed is very interesting, that the peptide bond is on this side, that means on the this is the C terminal and this is the N terminal. So, now this is a proline and this is usually an aromatic ring, a phenylalanine. What we know that like all the enzymes, they do not like to work or they cannot cleave the peptide bond if there is a proline. But here this is an enzyme which likes to have a proline and cleaves the bond between phenylalanine and proline. Okay. But that gives a very good handle, because that means if you can inhibit this protease, human enzymes, because they do not cleave the peptide bond involving proline, so they will not be affected much, because they will be entirely different character. Even if we have an aspartyl protease, but that is not able to hydrolyze the peptide bond involving a proline. Okay. So, that is why you can expect selectivity if you can inhibit this HIV protease. So, HIV protease, it can hydrolyze many other sites, but one of the primary site is this, that hydrolysis of between a phenylalanine and a proline of a glycoprotein. Okay. And based on the structure of this, the peptide, this is the functional, the protein, the glycoprotein which is hydrolyzed. So, what they have done, they have modeled different compounds and made HIV protease inhibitors. They started with a proline, because the enzyme, remember the enzyme is HIV protease which recognizes a proline and then hydrolyzes the amide bond. So, basically your compound should what? You start with a proline and a phenylalanine. Now, let us see whether L-phenyl L-proline, whether this is an inhibitor, or you increase the size, put different groups. So, by that way slowly the size of the peptide got increased, and finally what happened, you know that when the hydrolysis takes place, it goes via a tetrahedral intermediate. This was the case, na? If water comes, then this goes out, it is a tetrahedral carbon. So, you want to have a tetrahedral carbon at the site of the carbonyl, but it cannot be a carbonyl, because if it is a living, it has got a living group, then the attack will take place. So, what you have is one is what is called a transition state analog. So, you have a carbon with a OH, the sp3 carbon with OH, and then the carbon. So, this is a what is called peptidomimetic. It is not a peptide bond, but you are mimicking a peptide. But this peptidomimetic is basically based on transition state analog. So, they have put proline and then they changed the proline. Finally, they found that a fully hydrogenated isoquinoline is better than proline. Okay. So, we are not going into the details, it must be a trial and error, but they started with proline and L-phenylalanine, and finally, the drug that is now approved is called sequinavir. Sequinavir is a compound which is based, the design is based on what? Design is based on the that peptide which it hydrolyzes, there is a proline and there is a phenylalanine. So, it starts from there and slowly elaborated that. Only thing you have to remember that it has to use a transition state analog, you cannot use a peptide bond. So, that crucial peptide bond you have to replace by what is called a peptidomimetic. Okay. So, this is the first drug that was the HIV protease inhibitor. Then a success story from a Bengali scientist. Many people do not know this. Professor Arun Ghosh, okay. He was a student of Narendrapur Ramakrishna Mission College in Calcutta. Okay. Studied chemistry, went to IIT Kanpur, then went to Harvard, did his PhD, and then went to first to Illinois, and then now he is in Purdue University. So, he started also trying to develop HIV protease inhibitors, and again you see this was the carbon which is the peptidomimetic, that means the transition state analog, and he had the phenylalanine already there, but instead of the proline you have a sulfonamide now. Actually, this requires lot of in silico screening, that different molecules, how do they score when you dock with the HIV protease. Okay. So, ultimately he came up with a molecule which is called darunavir. If I am correct, this is basically let us see what is the configuration here of the phenylalanine 1, 2 and 3. So, that is S. Because what I know that this part comes from his name Arun. Okay. So, possibly there is some deconfiguration in the molecule, that is darunavir. So, he calls it darunavir and it is now approved as a drug, and so that is the contribution of an Indian scientist in developing an anti-HIV agent. Okay. Just to quickly, so this is the crystal structure how darunavir is interacting with the HIV protease. It is binding with, see, aspartate 25 and 25 prime. Here prime means one of the subunit and normal numbering is the other subunit. So, it binds with aspartate 25 and 25 prime, that is the key sites in the which does the hydrolysis, okay, which actually coordinates to the water molecule, and then in addition to that it has got other binding partners, that is why it is so, it works in nanomolar level. Okay. So, that is I think so. We have now discussed about how to develop antiviral drugs. You can target certain aspects of the virus life cycle, like you can target the if it is a retrovirus, reverse transcriptase, if it is a DNA virus, you can target the phosphorylation basically, and then you can inhibit the polymerase there. You can target the HIV protease in case of HIV. This is not for all other virus, but for HIV, this is HIV protease you can do, and this is important because the glycoproteins have to be synthesized by the virus. Just the last one, I think another strategy which was successful only in one case up till now is what is called antisense therapy. Antisense therapy is basically that you have the, if you have the, what is antisense? Antisense means the strand, the negative strand, which is copied to the sense strand. Okay. So, when the virus replicates, what it will do? It will make the sense strand of the mRNA. Okay. Now, if you know the sequence of the mRNA, what you can do, you can make the complementary strand of that RNA and give it to the system. So, what will happen? Suppose this is the sense strand of the mRNA. Outside, from outside you are giving, you are giving the complementary strand of the RNA. So, what will happen? The RNA will now form a double stranded RNA, or if I write it in linear fashion. So, if this is the sense strand, suppose this is the sense strand and I make a part of antisense part, suppose this. So, this will go and block the mRNA, form a double strand here like a primer. Okay. So, then what will happen? The translation will be stopped. This is what is called. So, that has to be antisense then, because you have to stop the sense strand of mRNA from working. Okay. However, there was a problem in this strategy. The problem was there are enzymes which are called nuclease enzymes, ribonuclease. So, when you give some RNA from outside, some RNA piece, that will be immediately chopped up by the RNA, the ribonuclease, the nuclease enzymes. So, before it binds to the target sense RNA, this whole RNA will be hydrolyzed. So, that will be ineffective. So, that was the major problem. So, what people did, very interestingly, they took the normal, see the sequence has to be the complementary. So, the normal bases have to be taken, but instead of phosphate, what they did, took the, this is there is a O here, this O. So, instead of phosphate, they have a thiophosphate. Now, this is what is called, also called phosphorothioate, linkage. So, basically what you have done, you have removed the oxygen, put a sulfur. You can say sulfophosphate, that is not a problem, but in the literature it says phosphorothioate. Some books have written phosphonothioate. I think that is wrong, because it was from a book. So, this is phosphorothioate, and that is the nuclease cannot hydrolyze this phosphodiester bond. Okay. So, these are called antisense with stable phosphorothioate linkage. That linkage has to be there, because in order to bypass the action of the nuclease, the ribonuclease, which is going to hydrolyze all these things. So, all these linkages are basically all phosphorothioate, and this is the drug. So, they have made this antisense drug for what? Cytomegalovirus. There is a virus called cytomegalovirus. Cytomegalovirus causes infection of the eye, very serious conjunctivitis in the eye. So, the strategy is to make an antisense oligonucleotide with a stable phosphorothioate linkage, and what happens when the virus wants to replicate, makes the mRNA, that mRNA is blocked by forming the double strand. So, that is the other strategy. So, there are basically vaccine is a strategy, then you have this a reverse transcriptase inhibitor, you have the DNA polymerase inhibitor, you have the protease inhibitor, and you have the antisense strategy. Besides, there are other strategies. I think for our purpose, knowing more or less the general strategies and the different drugs that are present or that are used today to treat viral infections. So, we have come a long way from 80s. If we think from 80s, we have come a long way in developing many of these antiviral agents. Okay, thank you.